Hello and welcome to Differential Discussions. I'm Melissa,
And I'm Dave.
and we're back this time with body fluid differentials.
Yeah, this is pretty exciting. You know, it's in the scope of what we do, but it's I don't know, tangential to the regular hematology stuff. Um, but yeah, I think body fluid differentials is always just really intimidating for people, especially first training. And I know that even still today, with all the experience that I have, you can look at a body fluid and it can be pretty spooky. So
Yeah, absolutely. So I guess let's start with the major difference is how they're made.
yeah, yeah, that's the big difference.
And so
So
most body fluids are thin, watery.
mm,
They're a lot thinner than blood and they're usually way less cellular.
yeah,
So in order to make sure that we are not scouring a blank slide for eons and eons, we have to concentrate all of those cellular products to one area. Excuse me. So we use a cytocentrifuge. And essentially, it's going to concentrate the cells to one small location on a slide.
hmm which is a huge benefit, right? But it does come at a cost. Yeah. So because of the cytocentrifugation of the cells, they kind of are high velocity hitting the glass slide and they kind of go splat a bit. So I think you're more prone to like smudge cells kind of things. And it's going to basically usually make the cell a little larger. And chromatin patterns become difficult too, right? Because they become exaggerated. So yeah. So yeah, we can kind of see in this field here a couple of smudge cells and some of that stretchy kind of behavior. Yeah.
I think the stretchy behavior is really noticeable in lymphocytes
Yeah.
because with the lymphocytes, they typically have a nucleoli, but they
Mm,
don't often show them to you in a peripheral blood.
right.
But in a body fluid, they almost don't have choice because of the slapping against the slide during the cytocentrifugation process, so they
Yeah.
spread and you can see that nucleoli.
Yeah. So the automatically every lymph is a blast, right? So that's
Yeah,
So there's
and
a
you can
little bit
even
of a
see that here.
correction.
This one's got a little nucleoli right there.
Yep. And just like a normal, possibly like semi low grade, like reactive kind of process, right? Totally normal physiology. But they can be a little intimidating, especially the newer techs and stuff.
Yeah.
Um, so yeah, part of like body fluid diff training is kind of desensitizing yourself a little bit. Um, so yeah, you have to have these lenses of frame of reference. Like I'm looking at a peripheral blood, and then I'm looking at a body fluid diff. It's gonna be different.
I think it's similar to when you're learning peripheral blood smears, and then you just have to get used to normal.
Yeah, true. Yeah. That's very true.
Normal, right? When
Mm,
we think about body fluids, they should have minimal numbers of cells.
yeah, like a normal effusion, right? Like essentially. So if we kind of think a bit about what we're talking about, so typically for body fluid collections, there almost always needs to be some kind of an effusion, right? Which is a pathological accumulation of fluid. So just by their nature, they're abnormal, but there's normal findings. So it'll be a little bit confusing as we kind of like talk through the dialogue a little bit. But yeah, so um, yeah, there'll be a lot of body fluid specific things that we kind of talk about here.
Yeah.
So
And we'll touch on some in this video, but I think a lot of them will be in the specific video. So as
right,
we're looking at CSFs,
right, right,
you
right.
know, bronchial alveolar lavages, as we're looking at serous fluids. So I think it'll be more specific when we're actually looking in the fluids, but we'll go over some today.
Yep, makes sense. Yeah. So start with like normal cells, right? So this is kind of our normal slide here. So Melissa pointed out a lymphocyte up in the corner there. Yep. And we have a few neutrophils to its right. And, for the most part, distinguishing um, lymphs and polys is sort of intuitive once you've mastered differentials. And I think one of the trickier cells is the mono max. So depending on the institution and procedure, we have a classification category that fits macrophages, which are fully matured tissue cells descended from our peripheral blood monocytes, right? So given that they have the same lineage, we're less concerned with splitting hairs on if something is a monocyte or a macrophage typically. And so we kind of lump those together. But those get a little bit tricky because there's the pleomorphic nature of the nucleus and the lots of the vacuoles, right? So even that foamy vacuolated appearance that's very common in body fluids is a little intimidating to kind of see, I think.
Well, and I think that when you look at some cells, it's easy to say that's a macrophage, that's a mono, right? Like you
Yeah,
can say this is a macrophage,
yeah,
it's a mono.
yeah,
They're clear, but sometimes you look at cells and you're like, is it a macrophage? Is it a mono? Does it really
yeah.
matter?
Yeah. So I've always been, as I developed as a professional, what's the clinical significance? What's the clinical significance, right? And because there's virtually none, then it shouldn't split that hair, right?
Yeah.
Um,
Varies by institution, but we won't split the hairs.
Fair enough, but I would love to debate whether procedurally it's beneficial to the clinical team and the patient to be kind of doing that.
I agree, but I do
Yeah.
know there's some institutions that separate.
Ooh, sorry. And I could be in an area of ignorance where I don't fully appreciate why it should be done, but cool. So yeah.
Why don't
Anything
we get
else?
into some of the more like body fluid specific either
Yeah,
findings, or I guess some of them are body fluid specific findings and some of them are you frequently see them in body fluids and very, very rarely in peripheral bloods.
fair enough, right? Yeah, I know exactly where you're going with that.
Yeah. Okay, so first here we have synovial lining cells.
Cool. So these cells would be specific to the source of a synovial fluid. And our synovial fluids, I'd probably say vast majority I've seen were knee fluids, and perhaps like what's the other one that's common is maybe like hip fluids and stuff. But
Wrist.
wrist is another one you'll see. But essentially, any place where there's opportunity for bone on bone, we have this evolved kind of system for lubricating those spaces to prevent wear. So these particular cells line the cavity of the joint, and they have multiple responsibilities. They are the barrier between peripheral circulation and the joint space. So they tend to create the fluid that becomes the effusion that we draw out of the joint, right? So that fluid is going to have nutrients, rich in nutrients for the cells in the area, and then specifically that lubricant, right, that I alluded to. So synovial fluid, when you first get it, typically has a high viscosity, and that's very beneficial for the practice. So synovial cells, we'll look at mesothelial cells later, but they're very similar in appearance. They have very similar functions. In my experience, synovial cells tend to have more blue to them. They're kind of a richer color, but I would say generally it's impossible to tell the difference. Or
It's
you
all
should.
about source.
Yeah, source. Yeah, exactly.
And then just to back up 1 second, where you were talking about the viscosity of synovial fluid, that's because of the hyaluronic acid that's present in there. And so when we're doing like cell counts, even sometimes when you're making the cytospin, you want to break down that hyaluronic acid. This way you can kind of have a more liquidy fluid and you can really get a better preparation of your slide. And of course, the only real way to get it into your chamber sometimes is without that hyaluronic acid in there. And then if you're running it through an analyzer for an automated cell count, you have to break down the hyaluronic acid. Otherwise, you're gunking up that analyzer.
Don't break my XN.
Seriously.
Yeah. So
Yeah, but Like you were
So,
saying, they're synovia sites. They're just synovial lining cells. So, but they look very similar to mesos. It's just source. So when you look at them, sometimes it's easy to look at them and think, is that a malignant cell? I'm not sure. And so, couple of things. One, you kind of want to see windows in between the cells and where they connect. If you're seeing multiple, so kind of like this here, where you can kind of see where they separate. Oh, yeah, and Dave's drawing it over here, so you can see where they're separating. That's really important, is to see the windows. And if you see them individual, they kind of look like this, where they have these little blebs. And yes, bleb is a scientific word. So if you see these little blebs, basically that's what helps them interlock with one another when they are in this kind of conformation all chunked together. And when they get ripped off, you see the little blebs like these guys here. And so that's characteristic of our lining cells.
good point. Yeah.
Other things like the nucleus, you should be able to see a distinct roundish nucleus.
Mhm.
A
Yeah.
lot of times when you're looking at a malignant cell, it's hard to see where the nucleus starts and where the cytoplasm ends, or it's hard to see where one cell starts and another ends. They all kind of blur together. So that's one distinction. I think the other big distinction is that you're looking at a joint fluid and you're not typically seeing malignant cells in a joint fluid.
Yeah, I would say I don't think I've ever, but I hate to rule out and say, never. I'm a never say
Yeah.
never kind of guy, I guess. But yeah, knee cancer is not as common as other malignancies out there.
It's just the nature of the fluid, again, where we
Yes.
have our joints. That's where that fluid is found, and you don't typically have malignant cells invading those spaces.
Yep.
So it's just not a common finding that we see in our synovial fluids.
Yep. All right.
I don't think that's right. Yeah, I guess the other thing is it's easy to confuse them with macrophages.
Yeah, definitely. Especially as the fluid gets older and older.
Yeah.
So if you have like an inflamed joint and some of these tissue cells are sloughing off into the fluid, and they sit around for a while, they become really like vacuolated, degenerated. Um, it's very easy to mistake them for macrophages, in fact. So, body fluids really are, you do your best. So,
Yeah, body fluids are kind of junky in general.
yeah,
Like you can see
definitely.
here, like look at all this junky debris.
Hmm.
Somebody might look at that and be like bacteria, but it's not. You typically bacteria is similar in size and shape, and they're also typically of a size where we can look at it and say, "Huh, it's bacteria." But these are two differences. Like, they're not similar enough. They're too wide in variation of size, I think is what I was trying to say.
Absolutely agree. Yeah. They should be somewhat uniform.
Yes.
Right? Yeah.
Yeah. And for the record, with body fluids, we'll talk about bacteria, but it's just bacteria. We don't talk about cocci and bacilli. That's
Leave
not
that to our buddies in the next lab, right?
yeah.
Yeah.
All right. Very similar to synovials, which we mentioned, are the mesothelial cells.
Yes. Yeah, yeah. So again, the nuclei are like really nicely rounded, maybe oval, but just they have a beautiful kind of contour to them, remarkably smooth. You can have multinucleated, right? So on the left side of the frame here, we have a mesothelial with 2 nuclei. Totally normal. Nothing wrong with that. Yep. If I were to see a cell that had 2 nuclei and the nuclei differentiated from each other significantly, that would be like a red flag for me, right? So one nucleus that's way larger than the other. But yeah, totally normal to see that. And we can see these blebs, right? So when we talked about the synovial fluid, if we look around the outside, we see those evidence of the tissue cells being like fused together.
This one shows the nice blubbing on it too.
Yep.
And really, like we were saying on the last slide, synovials, mesos, similar functionality, just different locations. So mesos are found in mostly your serous fluids.
Yep.
So your different cavities where in your basically your abdomen area here, where you're going to have the space around your lungs, the space around your heart, and the space around your basically your gut area. So
Mhm.
all of those, the pleural, peritoneal, pericardial, the P's, you find that's where you're going to find it, and then in the ascites fluid.
Yep.
I think the only other thing with mesos is really if you find an increased number of them, that can also be significant because of things like mesothelioma.
Mm. Mhm.
So if you're finding that you're looking at one of your serous fluids and you find an overabundance of mesos, that may be a reason to still leave it for pathology review because it could be indicating some kind of like mesothelioma type malignancy.
Yeah. High amount of mesos is I think my scrutiny level like kind of goes up on morphology and things like that. Try to be a little bit more sensitive. But yeah, this they're tricky. There's so lean on the resources that you have around you, more experienced techs and the pathologist for sure on body fluids. Yeah.
Well, and even worse is reactive mesos.
So with the reactive meso, we see the increased protein content in the cytoplasm kind of reflected in this like this blue kind of reactive picture. And yeah, they can look really intimidating. So yeah, we have a couple of instances here. Some of these I'd be deeply troubled with, to be honest with you, like in the lower right, like
Yes.
so like I don't know what's going on. But for the sake of showing like what a reactive meso,
Yeah.
um, this is a good demonstration.
Yeah, these colorations here.
Yep. Yep.
And you could find reactive mesos anytime there's inflammation, infection, malignancy, anytime there's some kind of inflammation,
Mm-hmm.
mesos can be reactive.
Yep. Yep. Yeah, so the textbook might describe them too as having a fried egg appearance. And I feel like when you have a reactive meso, it's like accentuated, right? Like that kind of
Kind of like this one here.
yeah. Mm-hmm. Yep.
Next up, bronchial lining cells.
Love these things. They're elusive.
They are.
So you actually tend to visualize bronchial cells on the chamber doing the cell count more often than you do in the diff in my experience. I don't know if they're just overly sensitive or what it is, but sometimes they don't actually survive the process. But this kind of tubular columnar kind of structure, if you're super lucky and it's like exciting, you get the wispy hairy,
The cilia.
thank you, I knew there was a word for it. So
Yeah, and
so
it's really just about they sit in the line in the bronchial area and they're kind of wiggling their little cilia and pushing along whatever mucus, bacteria, anything, just kind of pushing it away.
yeah, it's like basically like a broom, right? I mean, it's a biological cleaning system.
But seeing the cilia on the slide isn't very common, so a lot of times you'll see this.
Yeah. Mm-hmm.
I will say, when you do see the cilia, they remind me of in Pac-Man, the little ghosts, the little
Yeah.
ghost, with the little cilia at the bottom being their little tails. Yeah,
Yeah.
but yeah,
But yeah, if you don't get the cilia, that column, and then also I'll point out that the nucleus is kind of off to one side. That's like really, really super common.
yeah. I
And
mean, they're called ciliated columnar epi. So, right, if you look at them like they're ciliated, they look like a column. Makes
yep,
sense.
yep, yeah, we're really boring people when it comes to descriptions, naming conventions. So yeah, really cool. If it, on this, I just want to add too before we move on. If you see them on the chamber, sometimes if it's really, really fresh, you can actually see them like, they'll be dancing across the chamber. It's pretty cool.
Yeah, and depending on where you are, what institution you're at, you may note them. They're not usually part of the 100 though, because they're not a white cell.
Yeah,
But you may say it may be that they're present, it may be that they're few, moderate, many. It'll depend on where you are, but they're typically something that you know.
yeah. They have almost no consequence to them, right? Like they're just there or they're not.
Yeah. Oh, you know what? I wanted to remember to say is CSF lining cells, like the leptomeningeal cells. We don't have any images of them because they're even more elusive than these bronchial lining cells, but they look very similar to mesos and
Yes.
the synovia sites.
Yeah. It's kind of it's like form follows function, right? And it's just such a beautiful part of nature.
Yep. All right. So I think we talked about like the different cells that you're going to find in fluids.
Yes. Yep.
Now there's some other findings that you could find in a peripheral blood smear, but they're really rare,
Yeah. Mm-hmm.
like bacteria.
Yeah. Mm-hmm. I mean, I probably I can count on one hand how many times I've seen bacteria in peripheral blood. Even blood culture bottles that are popping positive, and visualizing it is really, really tricky. And usually, if you can, it's never been good, right? Like, so now conversely, when it comes to like septic body fluids, that's much more frequent to be able to visualize those things.
Yeah,
So
they're very easy to pick out. It's better if you can identify intracellular because
yeah.
then you know it was in the fluid long enough for the bacteria to be eaten,
Mm-hmm.
phagocytized. If it's extracellular, you never know if it's really a contaminant
Yep.
or if it's really in the fluid. So there's always that. But intracellular bacteria is always important, regardless of the fluid. So it doesn't
Mm-hmm.
matter if it's CSF, doesn't matter if it's synovial. The only one really is a BAL, so a bronchial alveolar lavage, because a lot of times it's contamination from normal mouth flora.
Mm-hmm. Yeah, the BAL is it's the only one that's technically not a body fluid, right? Meaning it's not physiologically um, produced by the body. So you literally are putting an instrument down deep into the branching system of the lung, deep into the alveolar, flushing with saline, pulling that back up. And so it's a sort of therapy. It cleans out a little bit, and then we can kind of do an analysis on it. But if you think about the lungs and the digestive tract, it's all outside the body, right? So very common to see some bacteria.
Yep. Yeah. And then it's important to note it if it's there. But again, we don't say there's cocci, there's bacilli, or there's coccobacilli, or anything else. We just say that there's bacteria.
I mean, most times when I see it in a joint that's supposed to be sterile, I run over to micro.
Yeah. Yeah. You check to see if micro already reported out their gram stain. If not, you run over and you get somebody and you say, "Hey, did you look at this yet?" No? Okay, can you come look at this for me? And then you confirm that it's bacteria with them.
That's why being there's a lot of value in being a
Yeah.
generalist and having diverse skills. And then I always loved having the specialization and then going to another specialized person to get that. Yeah.
Absolutely.
So,
So in line with bacteria is yeast.
yep. So beautiful images here. Some
Yeah, and so
intracellular
these are
yeast.
just budding yeast, intracellular budding yeast. There's no
Um,
hyphae here, but I think really for our purposes in hematology, we would just say yeast again. We're not going to specify whether it's budding or hyphae or whatever. That's more again for our folks in micro.
Yep. Leave it to the pros. But yeah, this can happen. I mean, especially I think yeast infections and fluids are generally like immunocompromised patients or under special circumstances. Bacteria tend to be much more fit.
Yeah.
But you see it with some degree of frequency, right? So
Yeah.
and we don't have pictures of the specific organism, but we had a patient at an institution Melissa and I worked at that had Cryptococcus neoformans in a CSF. And what's awesome about that is it's I'm going to be wrong and say the wrong things. Microbiologists go easy on me. But it's got like a mucoid
The capsule.
encapsulated capsule. Thank you. Yeah, all right. So I'm not crazy. So like the staining pattern and stuff is we actually had, like, when you look at the primary slide, you see this starburst effect and this negative area of staining, and it's
I actually
beautiful.
have a picture of it somewhere.
Woo! So maybe we'll do we can like tag that on at the end or something, do it afterwards. But yeah, really, really beautiful and rare finding.
Yeah.
But yeah, most yeasts represent like this. You see these you know.
Yeah. Little buds or little hyphaes.
Yep.
Okay.
So with the body fluid effusions, there, frequently will be bleeding, right? So you're losing red cells into the tissue space. It didn't leave the body, but it left where it's supposed to be. So it is a bleed. And so we'll have the kind of cleanup process that's important to remove these things, right? So these red cells don't belong here. So a lot of times we'll see something like the erythrophagocytosis, right, down in the corner. So monocyte macrophage eats the red cell. But as those things become digested, so when I lyse that red cell and I have all that hemoglobin and I start to break it down, we will get the iron, right? So once the iron's been removed, we get these blue-black granules. There might be other terms for this too, right? People might just say siderotic granules, I suppose. But yeah, really common, I would say.
Yep. And another finding that you could find in the peripheral blood is the
Sure.
erythrophagocytosis. Not really the blue-black granules,
Right.
but you I don't know that I think I may have seen it once.
Yeah, you see it every now and then. It's like it's an auto. I think I intuit that it's an autoimmune kind of situation, perhaps.
When you see it in the peripheral blood.
Peripheral blood, yeah,
Yeah.
yep.
Yeah.
Super common to see it in body
Yeah.
fluids, just because it's the literal normal. When you got that bruise, right? So it's just trying to take everything. You get that bruise and like it's changing colors and stuff. That's all because of your macrophages and everything doing their business, right? Macrophages are uniquely positioned to process broken down red cells and export the iron, right? There's a lot of really cool physiology associated with that. So yeah.
Are the cleanup crew.
They are.
Yeah.
Yep.
So that's usually when we'll see erythrophagocytosis. And just to break it down, phagocytosis, right? The eating of something. And usually it's a normal process where macrophages or neutrophils are phagocytizing like a bacteria, but erythrophagocytosis is the phagocytosis of red cells.
Mhm.
So
I was going to say, you could also glean a little bit about the age of the fluid by
mm,
the presence of some of these things too, right? So for blue-black granules to be present, it's probably been a fusion for a while, right? Like,
the bleed is old.
yeah, exactly.
Whereas if you have more red cells and then the red cells being phagocytized like here and here, then you're probably dealing with a fresher bleed.
Yep.
Yeah. 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 So another thing we know, typically they're present, or I don't know if maybe some institutions could say few, moderate, many, but typically it's a more of a qualitative finding.
I like that.
Similar, leucophagocytosis.
Woo! Yeah, so a lot of neutrophils show up to the party, and then geez, neutrophils don't last particularly long, huh, Melissa?
They
Yeah.
do their job and then they're removed.
Yep. So very common, right? So this is another, and then again, breaking the word down like you did, Melissa.
Yep.
We could see the root of
Yep.
phagocytosis. And then what is it phagocytizing?
Glucose.
Glucose.
White cells. The other thing is, like Dave said, this is a normal process, especially when you have inflammation or infection, and your neutrophils go out, do their thing, and then macrophages come and clean them up. Malignant cells also phagocytize other white cells and other malignant cells too. So leucophagocytosis could be a normal thing, or it could be a malignant thing.
Mm.
Right. Crystals.
So in this specific slide here, we have crystals of a joint origin. So there are times where you have a buildup of these chemicals, and they can no longer stay in solution. They crystallize and they cause a pathology in and of themselves. So we have these are just like perfect images, right?
So gout crystals are probably more well known, and then you have pseudo gout crystals. Gout crystals you don't typically see on a slide. You will see pseudo gouts if they're present.
Yep.
Yeah.
Yeah,
So
a lot of it has to do too with specimen collection and anticoagulant, right? So I believe like EDTA dissolves gout crystals and things. So
yep.
There are other preparations typically for doing a more conclusive kind of determination as to whether or not there are crystals, but we've seen them plenty of times on the cytospin.
Yeah. Yep. Absolutely. And so pseudo gout crystals, this isn't the way we identify pseudo gout crystals, but we can certainly see them. So Dave's pointing them out. You got a nice one there, nice one right next door. And this one has one here, one here, one here. Lots of gout crystals. And then this big, beautiful guy here. And even just looking at like the normal neutrophils, look at like you can really see the fine filaments in between the nuclei. Like you can really see them connecting to each other right here.
Yeah, it's actually really pretty. It's
Yeah.
something that you may not see as readily like in a peripheral blood. It's like accentuated in the.
Yeah. Like if you see a drawing of a neutrophil,
Yeah.
then everyone draws the filaments, but you almost never see the filaments. But body fluids, you can see the filaments. Yep. Okay, so that's pseudo gout. And then hematoidin crystals.
Beautiful. Yes. So, and we're seeing them in conjunction with blue-black granules here, right? So when we take that hemoglobin molecule, one of when we break it down, removal of the iron, that becomes the siderotic granule or the blue-black granule. And then the the porphyrin ring structure.
Yeah, the protoporphyrin ring.
Geez, oh my God, right? I have to surrender my SH. As that becomes metabolized, right, we get these crystalline kind of bilirubin. I mean, that's essentially what it is, right? And they're usually beautiful, vibrant, kind of yellow color to them. Really cool to see.
Yeah, these are the kind of crystals that you can see almost on 10x because they're so bright. So you might not see the crystal, but all of a sudden you see this bright orangey yellow thing and you drop down and then you can see the crystal.
Yeah. Yep. Yeah, really fun. Anytime you see these, it's like, ah.
Yeah. And some more, both pseudo gout and hematoidin crystals would be things that are qualitative in nature when we call them.
Yep. Oof, yeah. And malignancies, right?
Yeah.
So I would say like finding bacteria, yeast, and malignancy are like the big emergent kind of findings, right? That could be critical to a patient's well-being. So, to say, I wish we could give like this is what cancer looks like.
It varies.
It's so variable.
It varies by patient, by cancer type, especially the place of origin, right? So this is an adenocarcinoma. Where did it originate from? Is it lung? Is it prostate? Is it breast? It's all going to look slightly different depending on where it's originating from. And then each patient looks different. And I think common things, like I was mentioning with mesos, like it's hard to see the difference between the cytoplasm and the nucleus, where they start and end. It's difficult to see sometimes where cells start and end. Sometimes they overlap so much. Like if you look here, like is this one giant cell or is this a bunch of cells squished together? The other thing is the size. So they're usually humongous in relation to your normal cells, if you can see normal cells. So look at these red cells around here. Excuse me, look at these guys look like little normal white cells, and look at the size of these in comparison. Same thing with this little lymphocyte here with this in comparison. So they're usually very large, not always, but usually they're very large in comparison to your normal cells. And they're usually just, they look angry, they look deep, dark.
Mhm.
Like these guys are really dark and mean looking and "Urgh."
Yeah. So the one of the things, red flags for me, is when we talk about staining. So there's a lot of folks out there that'll stain something and get like, well, we'll take for example, the cell that I kind of am pointing out. They'll look at the staining and be like, I'm going to stain this again, right? And then and you get the same picture. It's the same cells, it's the same stain, and you get the reactivity. So I really want you to trust your eyes in what you're seeing, okay? You're QCing the slide as you look at it. So I can see like red cells. So when we think about things like right stain, there's multiple components to stain the more basic and acidic elements. And if they're behaving like they should in cells on the same slide, you have to believe what you're seeing, okay? So don't waste time hoping to get a different result and rerunning, right? And remaking the slide. When you see where it's difficult to make out where the nucleus begins and ends and the cytoplasm begins and ends, that is a red flag. You need to take that for what it is, okay? So that to me is straight to path, okay?
Yes. Yeah.
Um,
And like this one, you can't even tell. Like, is there a nucleus? Is the whole thing?
yeah,
Yes.
that should be deeply troubling, right? Like, um, every cell in the body, besides mature red cells, has a nucleus and a cytoplasm, right?
Mhm.
So, well, sorry, my poor platelets, my buddy platelets, but yeah. So we got some special edge cases, but generally speaking, they all have very similar structure, organelles, and things in there. So if they're giving you a look that you don't, truly recognize, I mean, you can even take. So there's a little bit of image compression going on here, but let's even look at the nucleus in this cell, right? Like, look how foggy it is between the beginning and ending of the nucleus. That's not normal.
Right. Yeah.
Other things I look for, red flag stuff, is so typically we have our genetic material, our DNA, inside of a nuclear membrane, okay? And that membrane maintains some level of organization. So when I see what I basically end up equating to like herniation of the genetic material through the nuclear membrane, that's abnormal, right? So these kind of excessive little vacuoles too can kind of be like a clue. So like this not definitive, but like I look for like the hole punch kind of stuff.
That in conjunction with all
Right,
the other stuff.
right. So that would be another piece on the pile. But like I'm channeling my like Burkitt's knowledge, right? Like so, you know, Google Burkitt's. Burkitt's is a very um,
Aggressive.
classic,
Yeah.
yeah, kind of presentation. So when sometimes I'll see body fluids that have that hole punch, crazy Burkitt's like look, really suspicious.
Yes. And the other thing is that a lot of times, not always, but a lot of times, malignancies are in clumps.
Yeah, yeah, great point.
So you can see here, there's clumps of these cells.
Yeah.
So when that's really where the scanning of your slide comes in, especially where you have a very finite area of the slide to scan. Scanning it's so important because you might, if you just drop down to your diff, you might not see the clumps.
Mhm.
But if you scan, then you could see the clumps of them around, and then you can make sure to include either them as part of your 100 or making a note saying that there are clumps of something present,
Mhm.
these large unidentified cells until we can identify them, and then straight to path. But
Yeah.
you need to make sure you know that they're there, that they're in clumps, maybe even where some of the clumps are on the scope. This way you can give it to the path and they know where to go and look for them.
Mhm. Yep, absolutely.
All right. Kind of
Yeah.
going hands in hand with
Oh, cool.
malignancies, signet rings.
Yeah, these signet cells. And in total truth, I don't know exactly what prompts them to be anywhere in particular. I think I tend to agree with where you were going, is like you kind of see these in malignant malignancies a lot. But I don't know that they're actually a malignant cell, right? So I think by and large, most of these types of cells are macrophages, normal macrophages, and they're just eating something. And then you get displaced. The nucleus just gets squished off to the side.
I think both.
Yeah.
I
Yeah.
think sometimes they're signet
Well,
cells and then they're associated with malignancy. And then I think sometimes it's just wonky looking macrophages.
yeah. 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 Yeah.
But it's important to note them because how do you know which one it
Yep.
is? I mean,
Yep.
if you're seeing other malignant cells, then well, yeah, you know exactly which one to kind of pass it off to. But if it's, if it's just signet rings is all you're seeing, that's still, I think, something that I would want to send to Path to look at and confirm.
Yeah, like I can't really fully describe why, but like this one terrifies me.
Well, I think that one is the sheer size, and if you look at that cell and the one to the right of it, they almost look malignanty to me.
Hmm.
Whereas this one, he just looks like a macrophage.
Yeah, so like on this slide, right, in this field, so in this field that I can see, I'm not as alarmed by
Yeah.
that presence. So again with diffs, it's all about reps. It's all about time. Okay. So like hopefully we can kind of help you, zero in on like the important features and things, but the more and more you look at the stuff, the more it just becomes like a reflexive pattern recognition, I guess.
Right.
Oh, cool. So so mitotic forms. Okay, so this is something that can be pathological or can be kind of normal. It's hard to determine, but you should always note it. Okay, so this is evidence of uh, reproducing cells. Okay, so while they could be normal tissue cells making other, healing, right, making other tissue cells, it can also be cancer. And I think by the organization of these, that they look pretty scary to me. I'm not like pleased with their presence. So
Yeah, I think it too. I'd want to see the rest of the slide because this guy here, he's either reactive or ugly looking. It's kind of hard to tell by looking at him, but this is either going to be some kind of weird macrophage or it's going to be something more insidious.
yeah, yeah, yeah. So mitotic forms, absolutely note it, and path. I think I more often than not.
Okay, so this is the Cryptococcus that Dave and I were talking about earlier.
Yeah. So this beautiful. If you see a pattern like this, it should raise alarm. I think specifically like this is a CSF finding, right, Melissa, for the most part.
Yep.
And typically people that are immunocompromised. I believe this particular patient was like a IV drug abuser, HIV positive patient. So all that tracks. But the biggest thing about the morphological features is that burst
Starburst, yeah.
here. So that's
Sunburst,
just,
starburst, whatever we call it. But it's you can see the organisms in there,
Mhm.
and then it has a really thick capsule around it. That's one of the big things when you're dealing with Cryptococcus is it has that huge capsule, and typically microbiology is going to do India ink, and you're going to see the black stain around it with the really big kind of buffering, pushing everything away from it, and that's the capsule. That's how you see it. But we can also see it on our body fluids when it's very rare and it's easy to miss. So it's not huge. It's like you can see the red cells around it. It's a little bit bigger than the red cells on here, but it's easy to just kind of be like, oh, it's stain, and just kind of ignore it and go on. So it is something you have to be cognizant to look for.
Yeah, that was such a huge moment for me as a tech, like to um yeah, and that was a great enriching experience. But just a little context, this will be super rare. I mean, but hopefully it's helpful.
Yep.
Oh, awesome. So we see I think there's like a couple of different terms that we can use here. I think most of us call them pycnotic cells, but I might be biased. I think in the literature, some of the literature, you'll hear them referenced as a necrotic cell. And I think really we're splitting hairs on what was the biological mechanism that produced the dead cell, because that's what these things are, right? They're dead. So as body fluids get older, they run out of nutrients, glucose, things like that. Everything's going to start to die off and eventually get these necrotic forms. But yeah.
So it's just the condensing of the nuclear chromatin pieces that are there. They kind of clump off and they'll eventually be removed. The cell might break open, but that's the nature of body fluids. You're kind of having these cells going in, doing their job, and then they're either being removed and phagocytized right away, or there's too many of them. They're overwhelming that reticuloendothelial system, those macrophages, and then the rest of the macrophages will come in at some point and remove them when they can. And so if they don't get to them, they become pycnotic and they die. It's just something to be cognizant of, because you want to make sure that you're not identifying random other things in fluids. You're not looking at it trying to figure out what else it is. And the other thing that it can help you with is identifying how old your specimen is. So if you're looking at like something that came out of some kind of tube that came out of a patient and sat in a collecting mechanism for a long time, then it's probably going to have lots of pycnotic cells. If you're looking at if you're looking at pus, it's going to look a lot like this too. We try not to count pus because there's no reason to count pus, but sometimes physicians insist we count pus. But if you do, you're just going to see lots of pycnotic cells. So it can indicate the age of your fluid and the age of all of these cells. Because if you have a lot of pycnotic cells, it's an old fluid.
And just for the clinical people that might stumble upon our videos, the reason why we don't want to count pus is because it won't be accurate. I mean, it just won't. The distribution of cells won't be even, right? The whatever system you're using. So if you happen to put one of these pussy fluids through an analyzer, it is going to be you're just getting random numbers. You might as well have Google roll a random number generator for your cell count. So we're not just trying to be difficult; it just literally won't provide an accurate result.
And if it's pus, it's just a bunch of white cells.
Yeah, if it's pus, then it's probably some kind of infection, right? And like you got your data.
Yeah.
So but yeah.
So I think for noting pycnotic cells, I think that depends on the institution.
Yeah.
If it's something that then it's just definitely going to be a qualitative finding because we don't count pycnotic cells. And it may be important if you have a specimen like pus where you just kind of put a note saying majority of cells are pycnotic and you're not actually going to give them a number or like a differential based off of it kind of thing. So it's more of a qualitative finding.
Yeah. The other thing I found too is with people that are maybe relatively inexperienced, a pycnotic neutrophil that has one nuclei will frequently be misidentified as a nucleated red cell. So
an RBC or a myelocyte.
Yeah.
One of
So
the two.
we see those kinds of mistakes too. So that's where it's like important that you can tell that it's pycnotic or necrotic so that it gets identified properly more than anything, I think. But yeah.
All right, and then the last two things are malignancies. So lymphoma, they kind of look like blasts.
Yeah.
Maybe large, funky shaped limbs.
Yeah.
With very prominent nuclei and very pleomorphic, abnormal looking nuclei.
Yeah. So a lot of whatever rules you're using to distinguish malignant cells in peripheral blood are going to apply in the body fluid, but it just has to be kind of noted the cytocentrifuge and what that's going to do and to distort the morphology. I don't find that it's usually a problem because it usually just makes them look scarier or more scary. So, it's tough for us to give you, hey, this is what a lymphoma cell can look like because lymphoma is such a diverse set of
very
disorders.
depending on the patient.
So it depends, right? So just employ your usual tools for detecting malignancy and things like that. But there's
Our
certain,
students will tell you that, like, we are our broken record for it depends. It depends on the patient.
and I struggle with that stuff too, right? Like of course it's an easy answer for me to give, but it's literally the correct answer.
Yeah.
I wish I could give more definitive stuff. It's reality.
Hematology is frequently gray.
Yeah, it is. Yeah. Art and science.
Yeah. And then blasts.
And blasts, a kind of broken record, right?
Yeah.
It's gonna be very variable based on what's going on. Yeah.
Yeah. Well, this is just kind of like our prefacing video, so that now when the rest of the body fluid videos come out, you'll have somewhere to go. It would be like, wait a second, what's a mesothelial cell? And we go over a little bit in our videos, but it's always good to have a place where you come to see what is, I don't know about normal, but what do typical findings in body fluids? Where are they? What do they look like? Sort of thing. And we hope you find this one helpful.
Absolutely.
So thank you for your time.
Thanks. Appreciate it.